Hello everyone, I am Puria Azadi and I am a PhD student at the University of British Columbia. Today I will present our recent work on a morphology-focused diffusion probabilistic model for synthesis of histopathology images. Histopathology is a diagnostic science that relies on visual examination of cellular and tissue characteristics in magnified tissue slides. Recently, high-throughput digital pathology scanners have been developed that can provide gigapixel high-resolution images of microscopic slides at objective magnifications of up to 40x. Majority of the research efforts have been on developing descriptive models with more emphasis on classification tasks. However, generative models can be used to create synthetic images and have numerous potential applications in pathology. They can provide near endless source of novel examples for educational purposes and proficiency testing applications. Also, they can provide data for training deep learning models in order to improve their performance. In this work, for the first time, we propose a diffusion model to generate synthetic histopathology images. Also, we utilize color normalization and perception prioritized weighting modules to learn morphological patterns. Finally, we conduct an extensive empirical study and a survey on the quality of the produced images. One of the main challenges related to histology images is the lack of consistency in the staining due to variances in the site-specific staining protocols. Color normalization strategies are able to boost histological discriminative models' performance. We propose employing the same framework to convert the input images to the same color domain in order to make the diffusion model focus on learning the morphological patterns, such as the cell shapes, density, and the distributions rather than their stain differences. Here is an example of the two images before and after color normalization. In the next steps, at first, noise, input image, and time step will be used for forward diffusion. Then time step T and genotypes will be embedded, and allowing the noisy image from time step T will be injected into our unit backbone model to finally produce image for the next time step. Then perception prioritized module assigns weights for time step T to focus more on morphological patterns instead of imperceptible features. Finally, diffusion loss function will be calculated for the final backpropagation. Here is an example of forward and backward diffusion process. In the forward generating path, by multiple small steps, high-quality images will be produced from complete noisy images. Now let's take a look at the produced images by our diffusion model and compare them with the images generated by progressive GAN. It is clear that ProGAN is not able to generate high-quality images in some samples. Some of these cases are shown in the red boxes. Also, we assessed various metrics and showed that our diffusion model could produce images with higher quality and better coverage of real data distribution. We also tried a survey from several expert pathologists on the quality of the produced images. Random real and generated images were shown to experts, and we asked them about their opinion, if the presented image is real or synthetic and how much confident they are. Finally, results showed that there is no statistically significant difference for their opinion on the quality of the real and generated images, and they are not able to distinguish the real and our model-produced histology images. Thank you all for your attention, and please refer to the paper using the following QR code for additional information.